Immuno-oncology encompasses a new era of medicine, focusing on the development and delivery of therapies that may improve the body's intrinsic potential for generating an effective immune response against cancer. Immunotherapies have been pursued since the 1990s and commonly involve priming or boosting the immune system, T-cell modulation, and immunosuppression in the tumor microenvironment. The success of these agents has augmented our understanding of cellular and molecular cancer immunology. Cancer cells express antigens called tumor-associated antigens, which the immune system can recognize and target. Such antigens are presented on the surface of tumor cells bound within a major histocompatibility class 1 or MHC1. This complex is then recognized by T cells, which interact via T cell receptors on their surface. T cell activation stimulates cells of the immune system to release granzymes and perforin, which trigger apoptotic signaling cascades to initiate the destruction of tumor cells expressing the antigen. Tumor antigens are also released from tumor cells undergoing apoptosis. Such antigens can then be ingested by dendritic cells, where they are processed and presented to T cells within an MHC1 and/or MHC2. Co-stimulatory receptors, such as CD28 on T cells, are also activated by B7-2 on dendritic cells, resulting in T cell priming, release of cytokines, and activation. The immunological response to cancer cells depends on a robust response by T cells that is specific for tumor-associated antigens. Monoclonal antibodies are designed to specifically target the tumor-specific antigen or tumor-associated antigen expressed on the cancer cell, allowing for immune-mediated tumor cell killing by engaging antibody-dependent cell-mediated cytotoxicity, complement-mediated cytotoxicity, and activating cellular phagocytosis. In the case of antibody-drug conjugates, ADC, which use the high binding specificity of monoclonal antibody, the ADC is internalized and the cytotoxic drug is released directly to the cancer cells. The killing of cancer cells can lead to the release of further tumor antigens, which can then further enhance the immune reaction against these malignant cells. However, with time, cancer cells can evolve. Mutations within their DNA can generate a selective advantage, which can include mechanisms to evade detection by the immune system. Various types of tumor cells have also been shown to express indoleamine 2,3 dioxygenase, or IDO, an enzyme with immunosuppressive properties. IDO has been correlated with an increase in the presence of a subset of T cells known as T regulatory cells, or Tregs. Tregs are potent immunosuppressive cells whose presence is elevated in many patients with certain forms of cancer. Further mechanisms to evade detection involve directly escaping T cell recognition by downregulating MHC1. Tumor cells also release cytokines such as TNF-alpha and TGF-beta. These cytokines suppress the activity of local immune cells to promote immunosuppression and angiogenesis, which promotes tumor survival. Tumor cells also overexpress inhibitory ligands and receptors, which normally regulate T cell responses, for example, increased CTLA-4 expression, and can reflect an immune resistance mechanism. This negatively regulates the early stages of T cell activation by down-modulating effector T cell activity and limiting IL-2 production, enhancing the immunosuppressive activity of Treg cells. Both CTLA-4 and PD-1 binding have similar negative effects on T cell activity. Tumor cells are also able to hijack the programmed death PD-1 immune checkpoint pathway, or upregulate glucocorticoid-induced TNF-related protein ligand, GITR-L expression to suppress immune control and evade immunosurveillance. With many tumors using these pathways for immune resistance, any immune checkpoint initiated by ligands could be blocked or modulated to prevent tumor immunity. New therapeutics are now being developed to do just this, with CTLA-4 antibodies and blockers of PD-1 indicating diverse opportunities to enhance anti-tumor immunity. Some of the most recent advances in anti-cancer immunotherapy include the ability to harness the immune system in the detection and targeting of tumor cells. Advances in targeted therapy include the development of bispecific T-cell engagers, or BiTE, antibody constructs. This technology utilizes the variable domains of two monoclonal antibodies. One of these two domains is specifically designed to bind to an antigen found on the surface of a tumor cell, whereas the other is designed to bind the CD3 on the surface of T cells. BiTEs are designed to act like a bridge between a tumor cell and a T cell. This interaction is thought to activate the T cell, which then releases molecules such as perforin and granzymes into the cytolytic synapse. The perforin molecules are then thought to assemble to form a pore in the surface of the tumor cell. This acts to destabilize the cytoplasm of the tumor cell, as well as act as an entry point for granzymes. Granzymes are serine proteases that can trigger an apoptotic signaling cascade which causes the death of the tumor cell. Also under investigation is the potential for oncolytic viruses to target and infect malignant cells, while leaving healthy cells relatively unaffected. The release of tumor antigens then potentially engages T cells to target other cancer cells matching the tumor-specific antigen profile. In addition, chimeric antigen receptor, CAR T cells, are modified outside the body to express CARs on their surface and then reintroduced back into the patient to promote T cell engagement with tumor cells. Tumor cells produce colony-stimulating factor 1, CSF1, a cytokine that regulates the infiltration and function of tumor-associated macrophages, TAMs, driving immune escape in the tumor microenvironment. Inhibition of CSF1 signaling by blockade of its receptor decreases the number of TAMs and reprograms remaining TAMs to support antigen presentation and bolster T cell activation. This therefore alleviates immune suppression and restrains tumor progression. The next step in tailored immunotherapy involves stimulating an immune response against the unique mutations, or neoepitopes, within each individual patient's tumor. This technology is used to activate tumor-specific T cells, which then locate and destroy tumor cells. Amgen is currently exploring strategies designed to augment the anti-tumor immune response while overcoming tumor immunosuppression. Amgen is committed to investigating the ability of the immune system to detect and target tumor cells. Understanding the inherent immune biology related to cancer may further guide strategies to modulate the human immune response against cancer.